Parker Howe, who did work with Dr. Rogers, evaluating the effect of acute and chronic interleukin-6 and interleukin-15 exposure in autophagy in cultured C2C12 myotubes.
All right, yeah, so thank you everyone. Appreciate the opportunity to present this project. I just want to give a quick recognition to Dr. Rogers for all of his help. Moving right along, this is a somewhat complicated title, so I'm just going to break things down piece by piece to help us all understand what this project is about. So first, looking at interleukin-6, asking the question, why should we even care about interleukin-6? Well, the link is with type 2 diabetes. We know that type 2 diabetes is a growing problem nationwide. You can see that there's a very large number of adult Missourians that have this disease. And the reason we focused on this in our project is because there's an established relationship between type 2 diabetes and obesity with chronic inflammation. Interleukin-6 is an inflammatory cytokine, and there has been previous research that has showed the IL-6 levels are chronically elevated in patients with type 2 diabetes. And then again, the other part of our project is looking at autophagy. Autophagy has also shown to be altered in type 2 diabetic patients. So our project is really looking at IL-6, and we are proposing that IL-6 could be part of the reason why autophagy is being altered in these patients. And then of course, conducting this research, ultimately, we hope to better understand these processes so that we can improve treatment outcomes in the future. So breaking things down a little bit further, looking at cultured C2, C12 myotubes, what are those? C2, C12 cells are a myoblast or muscle cell line derived from mice. They've been used extensively in the past and have proven to be a good model to study muscle cell metabolism. So it's a good model for us to use to answer this question. Looking a little bit more about what they look like as they are cultured. So they'll start in this myoblast form, and then by days 2, 4, and 6, you can see that they start to look more like what you would expect muscle cells to look like. That's when they're in their myotube form there. Keep this picture in mind. I'll be showing pictures later on that we got over the summer that look similar to this and how we analyze them. So the last thing to break down before I get to our results is autophagy, just mentioning briefly what that is. So if you look at the name, it means self-eating. It's a multi-step process where you form an autophagosome in a cell. And the idea behind this is that ultimately the cell is using it as a process to degrade cellular components for a variety of reasons that might include the recycling of metabolites or a few other things. It's a normal cellular process. We do know that it is upregulated in times of cellular stress. And as I already mentioned, there's a link between several disease states and altered autophagy, including type 2 diabetes. So now we can get to our results, looking specifically at what the link between interleukin-6 exposure is on autophagy in these cells. And hopefully you better understand what that question means now. So there's a few proteins that we looked at that we measured, P62, LC3B, and AMPK. In the process of autophagy, you would actually expect decreased levels of P62 with increased autophagy because P62 is degraded. And then conversely, you would actually expect increased levels of LC3B because it dissociates, it's not degraded. So with increased autophagy, you would expect more LC3. And then I'll mention AMPK in just a moment. Looking at the results that we were able to measure using Western blot analysis. So LC3, we did see a significant increase in its levels at these exposures and for this amount of time with IL-6. So this is a piece of evidence that's leading us to the conclusion that autophagy is potentially being modified by acute high doses of IL-6. With P62, we did not see any significant results. We did see a pattern begin to emerge, but nothing significant. It's something that would have to be teased out with further experiments, but these are some of our initial results. Now looking at the other protein I mentioned, AMPK. So this is a big complicated diagram. We just need to focus on these steps right here. AMPK, when it's phosphorylated, its activity is increased, and through that, it is acting as an inhibitor of a protein complex called mTOR. mTOR is an inhibitor of autophagy. So if you are inhibiting the inhibitor, you would actually expect increased autophagy. So with more phosphorylation of AMPK, you would come to the conclusion that you have increased levels of autophagy going on. So looking at our results again through Western blotting, we were able to measure a significant increase in the levels of phosphorylated AMPK in our cells with these exposures of IL-6 for the acute 3-hour time frame. So another piece of evidence that autophagy is potentially being modified by this cytokine. The last way that we were able to look at autophagy in our cells is by measuring cellular diameter. The thought there is that if you have increased autophagy or protein turnover, you would expect a decreased cell diameter. So the way that we measured cell diameter is that we would take 3 different measurements along a single cell, and then we would do that for all of the cells in our field here on the plate. Averaging that all together, we did see a significant decrease in cellular diameter with this exposure of IL-6. It's hard to tell with your naked eye, these differences, it's just on the micrometer level, but zooming in and making those measurements, we did see a decrease. So another piece of evidence that IL-6 exposure is modifying something with autophagy and protein cell turnover. These are all the results that we were able to generate over the summer. This project served as a really good starting point to potentially look at other questions and to lead to more future research down the road. So I just want to thank again Dr. Rogers for all of his mentorship on this project. I also wanted to thank Dr. Staudinger and the MKRC lab space. It's a huge, awesome opportunity that we have to be able to conduct this type of research here in Joplin. So I just wanted to thank him as well for providing that. Are there any questions?